Hello, everyone. Some of you may know me already, but for those who don't, my name is Jonsson, and I'm a Business Development Officer at UCLA Technology Development Group, TDG. Just as a full disclosure, I do manage most of the panel's IP, but they're really good and smart people. And so it's a pleasure to introduce the next panel, UCLA Fuels the Future, the journey from idea to enterprise. The panel is co-moderated by Heather Christofk and Aditya Bardia from UCLA Jonsson Comprehensive Cancer Center. Please give them a warm welcome.
Good afternoon, and welcome to this panel discussion, UCLA Fuels the Future, the journey from idea to enterprise. I'm Heather Christofk, and I'm the Associate Director of Basic and Translational Research for the Jonsson Comprehensive Cancer Center, and it's my pleasure to co-moderate this session along with Dr. Aditya Bardia. We are pleased to host this panel discussion on behalf of the UCLA Jonsson Comprehensive Cancer Center. Dr. Bardia and I have invited 4 outstanding Cancer Center members to share their experiences in IP generation and company formation with us here today. Dr. Bardia and I will take turns asking these expert panelists some pre-planned questions, and perhaps if we have time at the end, we'll take questions from the audience. So to kick things off, I'd like to ask each of our panelists to introduce themselves.
So I'm Tanya Stoyanova. I'm Associate Professor in Molecular and Medical Pharmacology here at UCLA. It's my great pleasure to be here. And what our research interest is and what we have been working on for the last quite a few years is developing new therapeutics for cancers, as well as we have projects on diagnostics. So it's really my pleasure to be here.
Hi, I'm Mike Jung. I'm a medicinal chemist in the Department of Chemistry and Biochemistry. And I have the pleasure of working with almost everyone on this panel on research for cancer and other diseases. We have a couple of drugs on the market for prostate cancer, and I hope to get others for other diseases. So I'm happy to tell you what I know about IP and how to form companies. So.
Hi, my name is Crooks. I'm a professor in the Department of Pathology and also Pediatrics. I'm a physician scientist working in hematopoietic stem cell transplantation clinically, and my lab works on how T cells develop in the thymus mostly, and it's very interested in seeing if we can develop T cells from iPS cells, the pluripotent stem cells, and SERM has been vital to this effort.
Hi, David Nathanson. I'm a professor of molecular medical pharmacology at UCLA. I run a brain tumor translational research lab that focuses on drug discovery and development for malignant brain tumors with a particular focus on glioblastoma.
Excellent. Hello, everyone. Good afternoon. I'm Aditya Bardia. I'm a medical oncologist and the director of Translational Research Integration at the Cancer Center. We're very excited about this panel discussion, and it's my pleasure to co-moderate this with Heather Christofk. So without further ado, we'll get going on our first question. I'd like to ask all panel members if they could share their experience about generating IP and starting companies from discovery made at UCLA. Maybe we'll start here, David.
I guess the first experience that I had was back when I was a postdoc in Caius Redu's lab, where we started a company called Trathera. And it was founded by Caius Redu, Johannes Chernin, Owen Whitty, Mike Jung, and myself. And the target that we were going after was an enzyme that was critical for the synthesis of nucleotides with applications in autoimmunity and cancer. And we developed a novel inhibitor against that kinase. That inhibitor has subsequently progressed into clinical development. And really, I think what that did was inspire me to think that you can actually do drug discovery and development in academia. In fact, in some cases, it might be preferred. And so in 2019, after starting my own lab, Mike Jung, Tim Clousey of Neuro-Oncology, and myself developed another company called Capmy Pharmaceuticals that was really aimed towards discovering and developing brain-penetrant drugs against glioblastoma. And I think importantly, what we've been able to do in both instances is translate these therapies into patients with these deadly diseases.
Our main IP actually happened as a bit of an accident, to tell you the truth. We were working on basic T-cell biology, and a couple of very talented people in my lab at the time, some nearly 10 years ago, Chris Seat and Amelie Montel-Hagen, were working on the culture systems that we had and discovered that by combining a few different things that we've been doing for years, they could differentiate T cells just like a thymus, but in the dish. And that led us to realize that we had the potential for an invention there that could have some implications in cell therapy. And ever since then, we've been working on that. But along the way, what I've noticed with that invention, if you like, was the way it fed our basic science back again. The way the model that we were developing for therapies has really enhanced the science in the lab as well.
As I said before, I'm a medicinal chemist, and I interact with biologists and MDs all the time. And usually what happens is a biologist and MD has some compound that they've either gotten it through a high-throughput screen or a tool compound that does something very important to them in their biology. But of course, they can't patent those compounds because they're already known. And so they come to me and say, Mike, can you make an analog? And the answer is, sure. Then I have to figure out how to make an analog. But we have to make analogs, and not just simple analogs, because in the patent world, there are two terms that have to be met. Your compound has to be novel and non-obvious. Novel is easy. It just has never been made before or described before. That's an easy thing. But non-obvious is a little bit tougher, because what is obvious to one person is not obvious to everyone else. And so a simple change in a molecule would usually not be able to be patented. So we have to come up with some significant change in the molecule without losing activity, of course. And I do that, my group. I don't do any of it. My group does all of that. And in fact, we've been lucky enough to be able to form several startups. In fact, the two that David mentioned, and one with Heather also. But so far in my career here, I've had 14 startups, one of which we sold for a very nice bit of money. And of the rest, 7 are still in existence and still moving forward. So I have 4 drugs in clinical trials, but the IP comes by taking known activity from known compounds, remaking the compound in a different way so that you get the same activity or better, but with something that's novel and can be patented. So I can answer other questions on IP at other points if necessary. Thank you.
And my experience at UCLA with IP, so one of the projects is actually, as Mike Jung described, we had a target that we validated biologically, where I'm a cancer biologist. But then what do you do with the target? So we thought that if anybody can make a small molecule inhibitor, that will be probably Mike Jung. So he made series of inhibitors, so that's one of the projects that we have been working on. Another project was on antibody-based therapies against cell surface receptor that we have IP on. That's a project that started during my postdoctoral fellowship at UCLA. That's the 13-year project. So it takes a lot of patience. And this is the project that now it's more ready to be moved forward with a collaborator here, Aditya Bardia, who has been helping us from the clinical side. So yeah, that's my experience with IP, and hopefully I'll soon be able to share other experiences.
That was great. I'd like to follow up a little bit on your experiences in launching companies and launching technologies, especially with Drs. Nathanson and Jung. What challenges did you face, and how did you overcome them? Why don't we start with Mike?
Money. Everything else is easy. The hard thing is, you have to have something that a person who, a VC, a capitalist wants to invest in. So you have to have something which is novel and good. You have to have good IP, but it all comes down to having the money to be able to do the preclinical trials, to start a company, to do the work that gets there. And that's hard. So what you do, of course, is pitch the idea to VCs over and over and over again. And every now and then, one of them weakens and says yes. And then you form a company and can move forward. But it really is the science here at UCLA. These folks up here do great science. It's not the science. You've got to sell it, And you've got to sell it to people that aren't easily giving up their money. But that's it. For me, money is the key.
Yeah, I agree completely with Mike. I think that's been the biggest challenge. With Katmai, we were dealing with, I think the largest obstacle was the indication, or is the indication itself. Glioblastoma does not have a high incidence annually, so it's around 10 to 15,000 patients diagnosed in the U.S. with glioblastoma. So you're already beginning with a relatively smaller market. And then the second part is the last therapy that has been approved for glioblastoma is the current standard of care, and that was in 2005. And so since then, there's been hundreds, not thousands, of clinical trials that have been tested in this disease, and they have just all failed. And so when you combine a low market and a high failure rate, you're having a lot of risk-averse investors. And I think the way that we've really approached that challenge is really the beauty of doing this work in academia is you can de-risk it here. You can further validate your molecule. You can further test where it's going to be most effective. You can optimize combinations. The academic setting has really provided us the opportunity to continue to develop this molecule and essentially mitigate that risk. That hasn't totally reduced the challenges about getting the money that we need, but it certainly has reduced the risk.
So just to build on this in terms of if you look at ingredients for success, based on your experience and maybe for each panel member, what do you feel are the essential ingredients for success, and how can you successfully form a company?
If we knew that, we would have successful companies.
Besides money.
Timing, I think, is absolutely critical. So you could develop a concept, as we did, and if it isn't the right time for that particular product, then it may fail because of the lack of money and the lack of either risk or just money that's in the system. So I think a lot of this is outside your control. You have to, what else would be the important, I think, is that you are willing to put the time in. It's extraordinarily time consuming to start a company, and it's only you and your other inventors who can really do the pitches to VC. So you have this full-time academic passion, and if you want to start a company, you have to realize it's not going to be secondary, but it's going to have a close partner with the time involved in the company.
You're absolutely right. It takes time and energy. You've got to pitch the idea. You've got to be willing to put in the time. And it's mostly convincing people that what you have is really worthwhile. That's the deal. Because people don't invest in things that they don't think are going to make them money. They're out to make money. They're not out to cure disease. We're out to cure disease. They're out to make money. And so you have to have some way of pitching it so that they get the idea that not only will it help cure disease, but it'll also make me a richer person. And that's not easy. It's hard to do. It takes a lot of time and effort, as Gay says. But you've got to put in that effort, or remember, nothing we do will ever help the world unless we can form a company and take it forward, or somebody licenses it and takes it forward. Otherwise, it's beautiful work that goes into a journal and you never hear from it again. So you've got to be able to go out and sell it to the world in order for it to make money, in order to help people.
And Dr. Jung, is there a specific pitch you feel works better, or things that are not that?
If I knew that, I'd have more companies. There's no particular pitch. Let me tell you, it's the biologists that do the pitching. I don't do any pitching. I'm there, and the people you're pitching to will say, "Can you make the compound?" I say, "Yes." That's it. That's all I ever do on these committees. But the pitching, you have to convince them that there's a need for it. There's some good reason. It can't be just the number 14th drug for some sort of breast cancer. It has to be something that excites them. That's maybe an unmet need in the market. And as David was saying, a small market is tougher to sell it in. Glio is a small market, so it's hard to get the money from VCs to push that forward. So do I know what to do in pitches? No. All I know is the biologists that do it and do it well are invaluable.
And I think I'll add, I think a really important aspect to consider is to make your science very differentiating. You don't want your IP or molecule that you're developing just to to be an incremental advance. I think it's got to be a real significant advance. So in addition to, obviously understanding where the clinical need is, having really good, tight science that is making a significant advance. And something I'll add that has certainly been the case with us is you got to have a good team. We've been fortunate with Mike to have a nice complementary team. We all serve different roles, but they're complementary roles. We all get along. We like each other, and that has been a real most of the time. That has been a real positive for us in terms of being able to move forward, particularly when you encounter the obvious roadblocks that are going to arise as you go through the journey.
Tanya, do you have anything to add? Ingredients for success?
I haven't started a company yet, but in terms of success, bringing something closer to that point, for me, it was first you have to be really excited about what you're doing and believe in it, because then you need collaborators to convince them to actually work on it. So the second, and it goes with the first one, is to form a collaborative team. So as I mentioned, we're cancer biologists, but we needed for one of the project antibody engineering expert, and for the other medicinal chemist, right here, Mike Jung. So, and then we need clinicians to really find out what will be the first indications, if you will, we'll go for. So for me, it's really to be excited, to have an exceptional team, and to have a focus and discipline to stick with the project, because it takes a long time. Some of these projects takes over 10 years. And the resilience, because none of this, in my experience, works from the first time. So there were a lot of failures at the end. You see one compound or one antibody, but there were many that didn't work. So in my mind, those are the key ingredients. I don't know whether for success, but those are the ones that are important to me.
Such good advice on this topic. Is there any advice you have for biomedical research trainees who aspire to generate their own cancer-related IP and perhaps start a company in the future?
Yeah, go for it. I think it's great. It takes guts. It's risky. It's a lot of hard work, but the benefits are fabulous. I mean, we have cancer drugs that came out of my lab that have treated more than 1 million men with prostate cancer. And that's a hell of a good thing to go to sleep at night thinking about it. But that would never happen unless you're willing to get in there and do the hard work and go for it. So I advise any biomedical researcher to go for it. Absolutely. Great idea.
But I still think that you want to learn the science really well first. Don't try and jump into it, during your undergrad years. Well, it depends what the product is. I guess you could do that with certain things. But a really good, solid foundation of science and research and understanding what the field is that you're working in and what some of the challenges are, I would strongly suggest that. The next thing is that if you develop this passion during, let's say, your PhD, that you discover something, is always remember to protect it. That would be my one message. One simple message is sooner rather than later, file an invention report. That it doesn't cost anything and it protects you, and you have to do it before you go public with any of your presentations or your papers. So that was something that was really drilled in during my experience, is how important that is. Again, as Mike said, or David said, that nobody's going to get cured unless you can commercialize it. And nothing gets commercialized unless there's intellectual property that's protected.
Yeah, I completely agree with what Gay and Mike were saying. I think you first got to stay rooted in the biology and the science, and make sure that what you're pursuing is a valid target. I think also really start to consider very early on the translational applications. What is the context that your IP or your drug is going to be used in? What's the patient population? How is it going to be combined? How's it going to be dosed? I think these are all things that need to be considered very early on. And one thing that certainly was helpful to me is start to develop, I guess you would say, interdisciplinary fluency. I'm a biologist by training, but I was kind of forced to learn chemistry, dust off my OChem books from eighth grade, ninth grade, and so that really forced me to learn all these other aspects. And you have to start to learn business. You have to start to learn clinical trial, how they're implicated, and I think these are really important characteristics to be able to pick up on, and I think what these guys said, it matters the most, and that is absolute persistence, passion, and dedication. There is just going to be so many obstacles that come in the way, and so you've got to be ready to fully commit to it.
I agree with everything that was said thus far. And for me, what was very important at the very beginning of my career to form collaborations with clinicians. Where do we fit what we're doing? Where is the unmet clinical need? And where would this fit? Another thing that I would advise trainees to learn a little bit early on is how to make a pitch. I'm far from being an expert, but I was put on a spot very early in my career by an engineer, a collaborator of mine. I'll tell you a funny story. So he had this chip that he developed. We had the biomarker and we had the clinician with clinical samples. And what connected the engineer and the clinician was me, the biologist. So I was told, you will do the pitch. So I put the slides, beautiful slides on the biology, even on the chip. And then I was told, no. Most of your slides need to be the market, the profit margin. I had no idea what that's all about. But so I learned in a week. It was, I didn't think I'll survive the pitch. I survived the pitch. We got the funding. It was funding just to validate findings. But I would advise trainees to really start thinking about that. And there is, TDG provides a pitch gym here that you can really get ready to do that.
Great. So now thinking of the future over the next 5-10 years, what are the major translational gaps that are preventing successful drug development, and where do you see the next major breakthrough emerging? We'll start with you, David.
Well, at least in cancer, it's resistance, plasticity, heterogeneity. Those seems to be some of the major obstacles that are presenting to us, whether it's targeted therapy, immunotherapy. So I think understanding how these resistance mechanisms develop, and then developing rational combinations to be able to overcome those mechanisms, I think that'll be really important. I think starting to design and develop drugs that are against targets that were previously determined to be undruggable. I think like one of the most contemporary examples that we're hearing a lot about is these new molecular glues that are targeting RAS, and they're just transforming drug discovery and development. And I think that molecules like this, antibody drug conjugates, I think this is where the field is going, and I think that that's going to be very exciting to see.
Well, I'm into the antibody directed conjugates, but to be honest, I'm a small molecule man. I like small molecules. They're easier to take. It just put a pill in your mouth and it's gone. And so I think that we need to continue, and hopefully with the advent of AI, that'll help drug discovery by making some of our decisions a little easier. I don't have any AI, I should point that out. I have OI, ordinary intelligence. So. But it's worked okay for about 50 years, so I'm going to keep going on it. Now, you were saying, what do you see in the next 5 or 10 years? I see sitting on a farm somewhere in Louisiana, watching out for the alligators eating our fish or something. But 10 years from now, I won't be here. These folks up here will be. But I hope that some of my students who are out in the audience will go on and make small molecule drugs into the future. What do we need? We need good testing. We always have. We can do nothing without biologists doing the testing. We can't run a structure-activity relationship without fabulous biology. And the one great thing about UCLA is we have extremely good biology. And we're on the same campus, right? To take a compound from a chemist to a biologist is a 5-minute walk. That's unbelievable. So what do I see in the next 5 years? Well, in the next year or two, I hope I see a drug from my lab. But in the next 10 years, I'm not sure what I'll see. Thanks.
It seems like ordinary intelligence has worked for you, Mike. Okay, our next question is, what role should physicians and scientists play in biotech formation? And how can institutions better support entrepreneurial investigators without compromising academic missions?
You're looking at me. That's a really complicated question. So I think you mentioned physicians in there, and it is absolutely essential that the sort of clinical perspective for an invention comes in early. They don't have to be the scientist working on it, but it is tremendously helpful to have some at least interaction, if not physician scientists themselves, working in the field, because they realize what are the most important questions and the most important challenges. So how an institution and a society allows that to happen is a very big question in this country right now, where we're losing physician scientists fast for various reasons. We have to find a way to support them, because I think otherwise there'll be a lot of opportunities missed. So I think, anything that we can do to encourage at least the interactions, if not individual physicians who are also driven by a passion for science to get involved is incredibly important.
So I do think that physicians and scientists go hand in hand. In my experience, everything that we have done in the space of translational oncology, if you will, we had a clinician, and I'm a basic scientist. So again, without a clinician, like really talking through where is the unmet clinical need, how does this fit into, because they're the ones communicating with patients on a daily basis. We're in the lab, I am in the lab, but they really know where all the discoveries can fit. So in my mind, they just go hand in hand. All of our projects were we had heavy involvement from physicians.
Oh, do you want me to answer? Oh yeah. I'll just echo this. The science is going to go nowhere without that partnership. But you got to have the basic science, but it can only take you so far. And then you really need to have that partnership with clinicians to be able to advance that science to commercialization, to the potential clinical application. So just echoing what Gay and Tanya said, we need to be fortifying these relationships and making sure that they persist in order to advance new discoveries and drugs into the clinic.
And one thing that Gay brought up, I should have mentioned earlier, this whole idea of IP, that we're always told you got to publish or perish. It should be patent, publish, and perish, because if you don't patent before you publish, you've lost the coverage of that invention, that molecule, whatever. So I give talks in my department on what do you do when you've got a great idea. You do the research, and then you patent it, then you publish it. It's important to do that, because otherwise no one will invest in your companies.
That's great. And remember the 3 Ps. That's great. In terms of when things are ready, in terms of tipping point, so what's your perspective in terms of when do you decide when a discovery should transition to a company versus continue work in the academic lab?
I'll jump in, shall I? I think the first thing that has to happen, like any good science, is that it's reproducible. And that is a huge problem within the field of science as a whole, is reproducibility. There are some very, very high-level things that get published in high-impact papers that, when they get transferred, if you like, to the commercialization, are surprisingly not reproducible. And so I think, for all of you thinking about this, is to make sure that the science is right before you start to get ahead of yourself. There's probably something there. There may be something even more interesting than what you were setting out to discover, but make sure it's right first. And that's important, obviously, for your academic career too. So it's, that's what I would say. And then if you're not clinical, is to then quickly form those connections that we were just talking about with clinicians to understand whether this is useful. It could be useful for something. It might be useful for something completely different that you hadn't realized. It may not be targeting the cancer, it may be reducing the side effects or something like that. But having good colleagues who are interested in what you're doing and who can convey that interest and excite or understand that what is exciting and what is novel, and then talk to your TDG, your tech transfer people, so that very quickly they can start to get involved and give you some context in terms of whether this is easy to develop, whether there are other partners on campus that you could talk to, like Mike, to make this a reality. And then after all of that, and after you've protected the idea, is then to start talking to the money people.
And in fact, it's not just that. There are certain things that you have to have that the money people look for in every case. The first is very strong IP. First of all, it's the activity. You have to have great activity that's reproducible, as Gay says, and that's important, that treats a disease, an unmet need, or something of that sort. You have to have incredibly good IP. They won't jump in if they don't have IP, don't have composition and matter on the compounds. You have to have in vivo data. They won't jump in and fund without good, strong in vivo data. And you need at least some good tox, maybe not full tox, but at least reasonable tox. And you have to know the mechanism of action of your compound, of exactly what's going on and why. And so there are 3 or 4 or 5 things that have to be lined up before you even start to pitch to a company. And as Tanya says, that's just the science. There's all of the business decisions behind it. For a pitch, you have to put together a business plan and show what are the competitors out on the market and how are you going to beat that? How are you going to make a part of the market? So when do you take it forward? When you have all of those things done. Otherwise, you do it in-house as long as you can with NIH grants or other grants to try to move it forward to get as much of the preclinical data as you can before you pitch. And so it's not easy, but there's a lot that you need to have done before you take it out of the university.
If I could just add something else, I was thinking as you were saying that, the patent, it's not enough to have a patent, an invention report. You then actually have to develop the patent. It takes a huge amount of work to develop the patent well, and the more complex the idea is, the more complex the patent probably. So really good patent lawyers are really important to get involved early too, and before you start going out to the VCs.
And we're lucky at UCLA because they use Foley Hoag, who's been our patent lawyers for a long time, and they're really good at writing patents. So those guys are fabulous. I'm glad that we use them.
Yeah, and I don't have experience yet with starting a company, but I was told you need to think about the product. How much would it cost to manufacture the product? What would it take to actually make a product from your invention? And what would be, how would you define it as better than what's out there, first in class, best in class? Would it have less toxicity or anything along this line? You have to distinguish yourself from what is already out there. In my case, I feel I'm never ready. It can be all more data, more stronger data. I was recently told I like to share our data internally with collaborators, and I was recently told on one of the projects, you're way beyond the discovery, you need to move this forward. And one of the collaborators who told me that, actually sitting right here, Aditya Bardia, looking at the data, getting all excited about that. So in my mind, that's what I was told it needs to happen before we know that we can move this forward.
I think we have some time for some audience questions. If anybody has a question they'd like to ask the panelists, I will walk you this microphone. Okay, while you're thinking of that, are there any other final thoughts from our panelists? Maybe, we've talked a lot about how scientists and physicians are rigorous about science, but have less skills for company formation. And just final thoughts on what skills are needed to transition to the mindset for engaging with the money people and getting your discovery launched.
I don't think I have any of the skills you need to do what you just said. I'm not a businessman. I never would be a business. If I tell you to buy stock, sell it, please, fast. So I'm the worst person to do that. I definitely depend on my colleagues to know more about the business end of things. And in fact, some of the people that I've dealt with have gone on to business school after to learn how to take care of things like this. But I don't think I will ever have the skills to actually do business presentations. So I wish I did, but I just don't.
I was told to stop thinking hypotheses and start thinking about milestones. It's not about testing hypotheses anymore. What does it take to really bring it forward, key experiments, and what are the experiments that will really make me believe that it should be moved forward or we should just drop it. So milestones, that's what I was advised.
And I would agree with Mike. I don't feel, not only do I not have the skills, I don't really want to do the CEO thing. So finding someone who is really good at that, once you've developed the idea and there's a patent in place and a company seems possible, I think teaming up with either a lawyer or a would-be CEO who has the right mindset and knows how to, do the pitch deck and so on with you. You still have to be involved in it, but having that expert there to hold your hand is very, very helpful.
Yeah, I agree. I think really leaning on your collaborators, the team that you assemble, because I don't think any of us are true business people. But you do have to learn a little bit, and I think one aspects that we've had to learn, some tough aspects, is you got to develop thick skin because you love your science, it's your baby, but you're going to get hammered and it's going to hurt. And so you just got to be able to be resilient, to be able to pop up and keep believing in what you're going to do. And so that's been the biggest learning experience for me, is to be really rigorous and thoughtful of your science, to not fall in love with it, to be critical, but also be willing to accept that criticism and try to improve upon what you've already developed, because that criticism might ultimately help you actually advance what your goals are, and that's to get your drugs or other IP into the clinic.
And that's one of the good things about pitches. Pitches are a pain in the butt. But the good thing about it is the comments that the people you're pitching to make. What about this? Have you thought of that? How do you know this? Have you reproduced it? That's important, because the next pitch would be much better, because now you've got some idea of what was deficient in what you tried to pitch earlier. So learning from pitches is important.
Questions from the audience?
With that team assembly that you all just mentioned, do you have any mechanisms to find those potential team members? Like, who do you go to? Is it all through tech transfer? Do you reach out to prior networks? How do you find those team members?
TDG was very helpful in that way. I wouldn't have known where to look, what rock to look under. But TDG connected us with someone who was actually a lawyer rather than the business side, but knew a lot from that angle. So I think there's various ways, and events like this is certainly a way, right? Certainly some of the meetings, most of the scientific meetings are attended by people in industry as well, and so you might have someone ask you as well.
Yes, I agree. And in the companies that I've founded, I didn't know the people. The TDG helped a lot in giving you the idea of who to talk to. Also, you talk to people about your science without showing them structures, but you talk about the science, and they can lead you to people that might be able to take a company forward. But it's hard to find. It's hard to find the right person to be the CFO, the CSO, the CEO. It's not trivial.
So maybe final question before we wrap up. You have
We've
experience.
stunned the audience into silence.
If you were to just summarize, your experience, it's a lot of hard work, it requires a lot of perseverance, resilience. But what has been satisfying or exciting in this whole process?
I mean, actually, the best thing is having an idea that you have in lab, really cultivating it, putting in the hard work to develop it, and then be able to see it translate into patients is the coolest thing that I've ever experienced. It is so awesome and so gratifying to actually for that to happen. And again, I think what we've been able to leverage is really use the academic setting to enable that discovery, to enable that development, get that out, get it to the experts who could then translate it. And then that for me has just been a remarkable experience and extremely rewarding.
When you're at a meeting talking about your science and some people come up afterwards and hug you and kiss you and say, you let my father live longer. Thank you very much for that. There ain't nothing better in this world, and that's even better than sex. It's unbelievable.
I know, we need to talk, Mike.
So just the idea that you've helped someone live a longer life, if you've made their lives easier, that's what we're all here on this earth for, is to try to help other people live better, healthier lives. And when you can do that with your science, it's wonderful. It's a wonderful thing.
I haven't had that wonderful experience, and I can imagine it is the best possible thing. And for both of you, put it another way. But no, how could there be anything more wonderful than what both of you have experienced? So I can't talk to that. What I would say on a sort of level, many steps before that, was it was really interesting actually to go through the process. I don't know how fast I would go through the process again, but at least once to actually see this other side of science and technology and medicine. I learned a lot, and so it was quite mind-expanding in that way, and I am very grateful to have had that opportunity.
I will echo that. I don't have the experience Mike has and David is experiencing now, but I can tell I'm as excited as them, just seeing the things that we do in the lab getting even closer to getting to patients. So I don't know how it will be another level of excitement if we actually are able to eventually bring them to patients.
And are there reasons why these discoveries that have made it to the clinic came out of an academic research lab here at UCLA versus a big company? What made it possible here?
Well, I think big companies are, they're worried about other questions. They're not able to switch from one to another. They're not really innovators. Biotechs have always been the innovative force in the pharmaceutical industry. You take something and you're willing to take risk and do things that others wouldn't do. Big companies would not do some of the research that we do because it's too risky. And so the major advances that we see over the years have come out of academic labs, and I think will into the future. As long as the NIH starts funding it again, we'll be okay.
Yeah, I'll add. It's all about de-risking, and I don't think that there is one biotech or pharma that is fully dedicated to treating glioblastoma, and typically pharma runs away from it. It's just too risky, and so in order for therapies that are actually going to be developed for a disease like glioblastoma, maybe other rare, highly lethal malignancies, it's going to have to be in the academic setting, because that affords you that ability to take on these high-risk projects that have a lot of educational and scientific value in addition to the potential clinical value.
That's a great point, and that's what we see in clinic as well, that most of these ideas do come from biotech, and then they are purchased by big pharma. Well, this was fantastic. We had an excellent panel, great discussion. Would like to thank the panel members for facilitating this fantastic discussion. Thanks so much.